Hello everyone, welcome to Biology 1124, Investigations in Introductory Biology. I'm Ed and I will be your TA for this semester. So before we get started on today's lecture, I just want to introduce myself. I'm Ed Higgins. I'm a PhD candidate in ecology and evolutionary biology. I do fieldwork and lab work to study freshwater microbial ecology. And so I focus a lot on freshwater mussels, which a lot of people don't really know about, but they're really awesome study organisms and they do a lot of cool stuff for the environment. So if you're interested in freshwater ecology or ecology in general, feel free to talk to me about it. So I live here in Norman with my fiance and the two incredibly adorable but very hyper dogs. So when you inevitably hear barking during some of these recordings or in our Zoom meetings, it's them. They get rowdy. This semester I will be available online 8 a.m. to noon on Mondays, Tuesdays, and Fridays. So we're going to try and keep Mondays for section 44, Tuesdays for section 58, and then Fridays will be kind of a free-for-all. These office hours will just mean that I'm at my computer. I'm probably going to have a Zoom room open, so you can just pop in, or you can email me and I'll make sure I'm ready. I don't know how instantly available I'll be, but I'll make sure to prioritize answering your guys' questions and making sure that you guys are taken care of. So what are we going to do in lab? The course is designed so that the lecture and the lab go hand in hand. So any concepts you learn in class, you're going to be applying in lab. And you're going to be doing this using scientific tools and methods and thinking in a very scientific way. We're going to design and perform experiments, collect data, and at the end of the semester, you'll be making a poster. And so many of you may not have designed a scientific poster before, but we're going to go over that in the future. So what's really important is that you're creative, you're collaborating with each other and myself, and that you guys have fun with it, because science is cool and we're going to have fun. So please look at the course policies. A lot of the questions that students have at the beginning of courses are going to just be answered in the syllabus and the course policies. These should be available on the lab canvas and probably also the lecture canvas, but I'm not sure. Read it thoroughly so you understand the policies, procedures, and schedule, because the schedule can be a little confusing for the lab. I'll be making sure to keep you guys updated on what we're doing. A lot of students have questions about what happens if you miss a lab. If you forget to turn in an assignment or if you do really poorly, what's really important is that you get a mulligan, you get a freebie. So if you miss a lab or you have an absence of any kind, you're just going to drop the lowest grade over the course of the semester. So if you attend every lab, then your lowest grade will still get dropped. It's just, it's kind of a catch-all. If you have any sort of reason for missing the week of lab, then it's okay, you're going to drop that grade. So don't worry too much about missing one. Assignments required for admission to lab doesn't really apply to the virtual sections because we're not going to meet in person, so don't worry about that one. The types of assignments we're going to complete, we're going to be doing lab quizzes at the start of lab on Top Hat, so we'll get more into that as the semester goes on. You're going to write up summaries of the week's investigations and create a graph of your data. You'll see the format for that on Canvas. And we're going to be doing at the end of the semester, as I mentioned before, a poster presentation via Zoom of an experiment you've completed this semester, just your favorite one. Again, this might sound a little intimidating, or maybe just not intimidating if you think a poster is fine, but we'll go over the details of how that assignment's going to work as it gets closer. So resources that are available on Canvas, there's going to be the lab manual, which will have tips for designing, graphing, and writing. This is going to be a really valuable resource, so make sure you have access to it and you can get to it on Canvas. And then you'll have investigation modules with the background for the lab, procedures, and tutorial videos. And so just keep in mind that this is fair game for lab quizzes. So some of the material that you're going to need to know will be found in these investigation modules. So lab safety in the time of COVID, what are we going to be doing to keep each other and ourselves safe? For the virtual sections of the lab, it's easy. We're not going in, so we don't have to worry as much about this, and I'm sure for the lecture components, you guys will be taken care of as well. So let's design a simple experiment to give you some practice. So as we go through this, make sure you're taking notes and keeping up with what we're talking about. All right, So this is an issue that's creating a lot of buzz among scientists. We're talking about bees. Thank you, Dr. Shaw, for that amazing pun. USDA research on honeybees is focusing on causes of population decline. You all might have heard that bee populations are declining, and it's pretty huge. So as scientists, we want to think about what factors can contribute to population decline. And so, take a couple seconds, think about what you think is going to be a huge source of these population declines. And most of the time that students think about bee declines could be caused by the introduction of a new predator. Invasive insects or invasive predators can come in and wipe out bee populations across the globe. Several years of hot, dry weather making flowers unavailable, so this is going to be linked to climate change, which is on a lot of biologists' minds. Environmental toxins can be a huge source, so pollution is what we're talking about. And then parasites and disease, of course, can cause population declines. And so more than just pollution, human disturbance of bee foraging sites, so deforestation and just development. As human populations increase and expand, we're going to influence the bee foraging sites. So these are kind of the directions that most people's mind goes when they think about bee population. So because this has been on a lot of people's minds, another direction that scientists have gone recently is looking at pesticides. And so we're looking at banning certain chemicals linked to bird and bee deaths. And so one target that has been identified has been chemicals called neonicotinoids. They're widely used as insecticide. They're derived from nicotine, hence the name neonicotinoid. And they affect the nervous system of insects in general. And so scientists are not sure what level is lethal to bees or if other factors play a role, but right now a big line of investigation is neonicotinoids. So how would you tell if the decreases in bee populations were due to neonicotinoids or some other factor? What question would we ask? So, a straightforward question is just, why are bee populations decreasing? We're noticing this destruction of the animals that are very vital to human life. I mean, they pollinate our crops and our food, so why are they declining? And what hypothesis could we test? So in this case, a hypothesis is a causal explanation. So it's not a casual explanation, which is how I read that the first time, every time I see the word, but it's a causal explanation. So we're looking at what the cause of the decline is. So in this case, the hypothesis that we would phrase would be exposure to neonicotinoids is causing the bee population to decrease. So going into a bit more depth, what does a causal explanation mean? When we talk about a causal explanation, we're meaning a statement that establishes a cause for the observed phenomenon. So the way you can phrase your hypotheses is using the word cause or because. Bee populations have declined because of insecticide exposure. So when you're forming your hypotheses, you need to be thinking about, well, here we have our question, what's causing the source of the question? So students sometimes have been taught that if-then statements are good hypotheses. So if bees are exposed to the insecticide, then they will have lower body weights. And so we're thinking about body weights because that's a good indication of health in a lot of organisms. But if-then statements are really closer to a prediction. You're thinking about the future events, so you're predicting what's going to happen next. And so we're going to try and distinguish between future predictions and hypotheses. So just keep in mind that if you're thinking about the future and what's going to happen if you do something, then that's a prediction rather than. So if we're thinking about our hypothesis about neonicotinoids causing declines in bee populations, how did scientists go about testing these hypotheses, or this hypothesis rather? Scientists determined that bee body weight may be a good indicator of health, and if you have sick bees, then your population is going to decline. And this is common in a lot of organisms. Body weight can often be linked to health. So these scientists tracked and measured the weight of bees that were found foraging at 25 different locations within a country, and then they sampled the amount of insecticide residue at each of the 25 locations. And some locations had higher residue concentrations than others. So if we're talking about a scientific experiment and testing a hypothesis, we're going to have to talk about variables. So the dependent and independent variables are the ones we're going to be talking about. So the dependent variable is the measured variable, or what you're trying to find out. In this case, it's the weight of the bees. And the dependent variable is always dependent on the independent variable, or it's influenced by the independent variable. So in this case, the body weight of the bees in our hypothesis is affected by the neonicotinoids, or it's dependent on the amount of insecticide being used, so it's the dependent variable. The independent variable, on the other hand, is the manipulated variable, or the part of the experiment that's different between the sample groups. So while these scientists didn't actually manipulate the amount of insecticide used, it is different among the sites. And the amount of insecticide applied isn't affected by the weight of the bees in the area, but the weight of the bees in the area might be affected by the amount of insecticide. So when you're trying to figure out which is the dependent and independent variable, think about what variable you're measuring to see the effect on another one, or is going to be the independent variable. So once you've gone out and you've collected all of your data, a critical component of actually understanding what's going on is graphing the data. You can take a bunch of numbers and you just look at the numbers and it doesn't tell you anything. So making a chart or graph is a good way to see what's actually happening. And so in order to do that, you need to identify your variables. Again, the dependent variable depending on the independent variable, and you put the independent variable on the x-axis and the dependent variable on the y-axis in most cases. So in Excel, which is the software we're going to be using a lot in this class. You can get it for free from the OUIT website, so just make sure you have Excel and start familiarizing yourself with it before you start trying to manipulate the data. So in Excel, data for the x-axis is going to be placed in the leftmost column when you're making these. After you've collected and identified your variables, you're going to then have to decide the right type of graph or chart. And so bar charts are good for comparing categorical data, so things that are divided into categories or groups, whereas line graphs are good for correlating continuous variables or comparing change over time. So if you have any questions about the difference between categorical and continuous data, you can feel free to ask me. Taking a look at a bar chart here, can you determine the relationship between the two variables in this graph? So if the scientists here, they gave different servings of ketchup or measured the different servings of ketchup that people consumed, and then they compared it to the weight gained. So looking at this chart, you can kind of maybe see some ups and downs, but personally, I can't really tell what the relationship between the two measured variables were here. So you might think that because the servings of ketchup eaten and the weight gained are both continuous variables, that converting it to a line graph might work. But in this case, while you can see some sort of relationship between the up and down of the top and bottom graph, these two sets of data are just thrown onto a graph and trying to see what happens. But you can't really see the relationship between the two variables of the group, so we have to try a different method of plotting. And so here, we can see when you plot the servings of ketchup, the independent variable, so the variable that was manipulated, and on the x-axis, and then you put the weight gained or the dependent variable on the y-axis, you can see the relationship between the how much ketchup was eaten and the weight gained. And with this equation, so if you remember y equals mx plus b, you can actually calculate if I have 2 servings of ketchup per week, how much weight can I expect to gain? So this is going to be the kind of graph that you'll need to make to actually see the relationship between these variables. So for this week's assignment, you're going to use the supplied data in the Excel file on Canvas to create a graph in a short video. So as I mentioned before, we use Excel in this class, and it's a free download for students from the OUIT webpage. So if you don't have it, don't worry, just go to the webpage and you'll be able to download it. To actually complete the assignment, if you're running into some struggles, just follow the instructions in the graphing and video assignment instructions file available in the first week module on the Lab Canvas page. So you may run into some problems as you go, even with the instructions. So feel free to use Google and YouTube and try and find other resources to help you learn Excel and figure out how to make these graphs. Inevitably, that will not help some weeks. So when you do run into issues trying to figure out what you need to do, come to me. I'm a good resource. I have the office hours available, and if you email me, I'll be able to answer questions that you have. And so this assignment, you'll have a week, and you'll just have to turn it in by Friday at noon. So just a word of caution, try not to wait until Friday morning to submit your assignments. We're all going to be very busy this semester and it's going to be somewhat hectic. So it's understandable that you kind of push things off till the last minute. But with these, if you run into an issue and it takes you longer than you expected, and then the office hours are full and you can't quite get your question in, you may not be able to complete the assignment on time. So try and, open it and try and take a few stabs earlier in the week, and then, we all procrastinate, but just try not to do it too much. And again, if you have any questions, feel free to talk to me, and good luck this week.